Okay, so for this lecture, we're going to look at different types of proteins. So this lecture will be useful in maybe helping you select a pet protein for you to do the activities on the pet protein for Unit 1 and Unit 2. So we're going to look at several different types. So the first group we're going to look at are enzymes. So enzymes are going to help form products, so they're going to catalyze reactions. So they help to bring substrates together, and because they're bringing these substrates together, it's more likely for the reactions to occur. So if you remember back to like an introbio class or even a chem class, you would have talked about that you have substrates and then you get products. And what helps that occur by decreasing activation energy would be enzymes. Okay, so enzymes play a role in a number of different biochemical pathways. So our metabolism, being able to take sugars and convert those into ATP, usable energy within the cell. Or if we look at production taking photosynthesis and taking carbon dioxide and making sugars, there are enzymes that happen with that. If we look at when we eat food, pepsin helps to break down the proteins in our stomach. And so again, these are all examples of different enzymes, even DNA polymerase. If we think about DNA polymerase, it's going to bind to DNA. It's going to bring all the little components that we need, nucleic acids in, so that we can make copies of the DNA. So for our next group, we're going to be looking at structural proteins. So structural proteins are going to allow for there to be support in the cell. So if we think about the cytoskeleton of a cell, those different proteins that help to form that scaffolding that's going to give the shape of the cell. Or if we think about like a cell that's going to change its shape, like an amoeba, it has to be able to rearrange its cytoskeleton in order to do that. So structural proteins are very dynamic and they change due to stimuli. So some examples of structural proteins would be like collagen or elastin. So these can form fibers in our tendons and ligaments. If we look inside cells, things like tubulin, which forms microtubules, or actin that forms filaments. Actin's really important for that rearranging of the cytoskeleton so that we get things like amoebas that can move, right? But we also have keratin, so that's going to help to reinforce epithelial cells. If you go into the grocery store and look at hair care products, keratin is like one of those things that they want to add into all these products because it's meant to help fortify your hair and such. So our next group of proteins are transport proteins. So they're going to help to carry small molecules and ions, either like in our body or within our cell, or out from outside of our cell into our cell. Okay, so an example of this would be like albumin. Albumin is a protein that's found in our blood, in our serum, and it helps to carry lipids in our bloodstream. Also hemoglobin, so a lot of you probably heard about hemoglobin, and so that's going to be transporting oxygen. So that's found within red blood cells. Transferrin is going to help to transport iron through our blood. So there's also going to be transport proteins that are embedded in the cell membrane. So in chapter 11, when we look at the cell membrane, we'll look at the role of some of these transport proteins. And so there are transport proteins that are important for being able to transport hydrogen across out of the cell and into the cell. And both in eukaryotic and prokaryotic cells, so both bacteria cells and also our own cells are going to have these pumps that will transport hydrogen in and out of the cell, pumping it out to create that concentration gradient that's used during the electron transport system. And we also have glucose carriers, so we want to make sure we're bringing glucose in so that we can then through cellular respiration metabolize that glucose. But we need to transport it in first. So right, we eat, we break that down, but then we have all this glucose available. We need to be able to transport that in. Then if we look at something like muscle contractions, being able to have calcium pumps are really important because that's going to allow those actin filaments to contract, allowing us to have a muscle contraction. So our next group of proteins are motor proteins. They're going to be responsible for movement in the cells or tissues. So if we look at like a skeletal muscle, we have actin that's going to give our structural form. We have our calcium pumps, which are going to allow for transport of the ions, and they're going to work together with like myosin to allow for movement, contraction of those muscles. Okay, with inside their cells, there's also examples of like dynein, that's going to allow for cilia and flagella to beat and move, so allow for movement of these eukaryotic or prokaryotic cells. So our next group are storage proteins. So storage proteins allow for things like amino acids and ions to be stored. So they'll bind them so that they then can be utilized later on. So several examples of storage proteins. Ferritin is able to bind iron so that it can be stored. Ovalbumin, which is found in egg whites, is where amino acids are actually going to be stored so that a developing bird embryo inside that egg can utilize that egg white as a food source. Casein, which is in milk, is then a source of, it's a protein that's found in milk, and so it's a source of amino acids when mammals feed on milk. We then have signaling proteins. So signaling proteins are going to allow for stimuli from external to become signals inside the cell. So it's a way for cells to communicate to each other, what's going on. And we're going to talk more about cell signaling later on in this semester. So just to kind of give you some examples, there are hormones that are going to be amino acid protein based, like growth factors, that will cause physiological functions within an animal. So examples would be like insulin. So if you've taken like an A&P class, anatomy physiology, you might have talked about glucose levels and how those are regulated. So insulin allows for the regulation of that. We can also have like growth factors, like nerve growth factors that allow for nerve cells to grow axons, or epidermal growth factor that causes growth and division of epithelial cells. So again, these all would be important proteins and that cell signaling in these different types of cells. So our next group are receptor proteins. So they're going to work with those signaling proteins in order for some kind of change to happen, whether this is a change at the organism level or whether this is a change at the cellular level. Okay, so they're going to detect signals and then transport them to the cell so that there can be a response. So rhodopsin is a protein that's going to actually detect light, and so that would then allow for your eye to respond to light. Okay. Also, we talked about insulin, but there's going to be an insulin receptor, so that insulin receptor is going to respond to the insulin in order to have glucose uptake. Okay. And lots of other examples of receptor proteins, and there's going to be receptor proteins on viruses and bacteria that allow for, if we think about viruses, the receptors that are going to allow viruses to enter into the cell. So if we think about coronavirus, that is going to be the ACE2 receptor that's going to allow for the coronavirus to enter into like tough cells and other cells in your body that have this ACE2 receptor. So our next group of proteins are the gene regulatory proteins. So these are going to function by binding to DNA to switch genes on or off. So these regulatory proteins could either be blocking the site where DNA polymerase or RNA polymerase are going to bind to. And so if they're there, you can't get replication, or you can't get production of RNA. So you wouldn't get the protein, you wouldn't get a downstream effect. Or in some cases, these gene regulatory proteins, when they bind to the DNA, it makes it easier for the DNA polymerase or the RNA polymerase to bind. So then you would actually get a stronger response. So it all depends on whether it's a repressor or an activator, what effect it's going to have. But in both cases, they're gene regulatory proteins. So an example would be lactulose repressor. So in this case, when it binds in bacteria to the genes, it actually stops the production of enzymes that are required for degrading lactulose, which is a sugar. So, I mean, if we think about this, if there's not enough lactulose around, you don't want to be making enzymes to break it down because there's no lactulose around to break down, so it's kind of wasted energy. And so by having this lactulose repressor bind to the genes, blocking it, you're not going to get the enzymes being produced. Now, once lactulose is present, the repressor comes off, and so now you can make these enzymes that can degrade lactulose, and that's energy well spent because you now have lactulose present. You need to break it down, you need to metabolize it because you're a bacteria. Another example would be the homodimer homodimer proteins. So these are also referred to as the Hox genes. So the Hox genes code for the homodimer proteins, and these proteins will bind to different genes that are important in the development, especially in fetal development of multicellular organisms. So genes like the hedgehog gene are controlled by these Hox genes. And then lastly, we kind of have this like miscellaneous category, special purpose proteins, and so their functions can be really diverse depending upon, and again, it's kind of this miscellaneous category. We really can't put you in one of the other categories. So certain organisms can produce like antifreeze proteins. So there's different fishes and frogs that produce these antifreeze proteins so that their blood doesn't freeze when the temperature drops low. So obviously if you live in the Arctic or Antarctic or even, in the northern states, it gets really cold during the winter. So if you're a frog out there, you don't want your blood freezing and crystallizing because that can damage your cells and you'd probably die. So by producing a little of this antifreeze protein, having it circulate in your blood, it prevents your blood from freezing, just like antifreeze in a car. Okay. We also have the green fluorescent protein that jellyfish can produce, creates a green light. Most of us, or in the near future, you will probably definitely hear of green fluorescent proteins. And so GFP, so we as scientists, as biologists, have utilized the ability for this protein to produce light. We now have that gene, so it can be expressed in like bacteria, but really in any kind of cell line that you have, you can insert GFP, green fluorescent protein, to a gene, and then when that gene is being produced, it will actually produce green fluorescent protein, or GFP, that you can use a microscope in order to look at that. And we'll probably look at that technique as one of our techniques in our tech talks that we'll do with each unit, but it actually came from jellyfish. So, and again, there's a lot of other miscellaneous. There's like a glue protein that allows for marine organisms to attach to rocks. So if we think of like coral, they need to be able to firmly attach those rocks so they don't actually get washed away by the seawater, by the tide. So any of your tidal microorganisms would produce this glue protein. So hopefully this gives you an overview of some of the different types of proteins. If there's a category of these proteins that you're interested in, I definitely think that you should potentially do one of those for your pet proteins. So, whenever there's an assignment, it's always best to pick something you're interested in. Each one of these different types are very interesting for different reasons. So if you need any help picking your pet protein, just drop me an email. Tell me a little bit about yourself and what you're interested in, maybe what your career goals are, and I can, send some ideas your way so that you're interested in doing. It's going to be the pet proteins, a 2-part assignment, and then we'll move into cells. So ideally what you'll be doing is you'll look at a protein for the first half of the semester, and then the second half of the semester you'll then look at the cell that produces that protein. Okay, so the 2 remaining pet assignments for Unit 3 and Unit 4 will be focused on a cell and how the cell responds. But again, those pets can be linked together. Okay. All right, thank you very much.